acids. They're getting connected up into a chain of amino acids. I have this animation that you'll be able to see in a second that actually shows this process a lot better. We'll watch that in a second. All right. Another vocab word that you need to know. Once again, I know there's a lot of vocab, but definitely go back, rewatch this video, review your notes to know what the vocab is. But once we have chains of amino acids, we form bonds either between them, so we connect the chains. Those connections between them are called peptide bonds. And these are just the connections between all the different amino acids. So these are all amino acids we're connecting together by forming those peptide bonds. All right, so here's a quick animation actually showing this process happening. So we have our mRNA, this orange square right here, rectangle, is the ribosome. And we have the tRNA, which read the AUG and brought in the methionine. And watch what happens. The ribosome moves along. It now is reading CCC. The tRNA brings in a proline. The peptide bond forms. And now the ribosome moves on. It reads AGU. It knows to bring the glycine amino acid. That peptide bond forms, and it moves along. It reads GGU. Okay, bring me a valine, peptide bond forms, it moves on. And then once it gets to the very end, we now have our protein, our chain of amino acids, and this is the process of translation. Okay. The last step, step 6, is the tRNA reaches that stop codon. So there's, just like we had one start codon, AUG, we have 3 stop codons, UAG, UAA, and UGA. This is how the ribosome knows, okay, we are done. We've made our amino acid, we've finished our protein. We are done making more protein. And here's a finished product of our protein. It's kind of all folded up, but this is what a protein might look like. All right. And a protein is basically just that chain of amino acids. And they can be folded into a very specific shape, which is this one right here. Here's my next couple. Yes?
Can you hear it? Yeah. And skinny and with the
All right, so let's watch a quick animation of this happening. This is a really cool animation because it kind of shows the whole process of translation in action. So right here, remember this yellow mRNA? Our mRNA is now leaving the nucleus. This is the nucleus, it's kind of coming outside the nucleus. And that mRNA is now looking for a ribosome. Let's see if we can find a ribosome. Oh, here's our ribosome. This blue part right here is part of the ribosome. This purple part is the other part of the ribosome. And you kind of see these things that are flying around, like almost like butterflies. These are the tRNA. We have lots of different types of tRNA. They have this little pink part at the end. Those are the amino acids attached to the tRNA. The tRNA is going to kind of bump into, here's actually, you can see this is the amino acid attached to the tRNA. It's going to find the right part of the mRNA. So notice how the mRNA is kind of moving through the ribosome. The tRNA is coming in, bringing in the right amino acid. Those amino acids are kind of connecting up to make a chain of amino acids. And so it happens really fast. Notice, just like the other one, it's happening bam, 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 bam. It's happening very quickly, bringing in those amino acids, forming those peptide bonds, and getting that big chain of proteins. Okay? This is actually happening inside all of the cells of our body right now. Proteins are being made throughout every single cell in our body, which is kind of a trippy thought, right? Like, this is happening as we watch this. Like, we wouldn't be able to watch this if we weren't able to have this happen. I don't know. Get all meta with it. All right, so let's talk about the key differences between the two key words we learned, transcription and translation. The first big difference, transcription is taking DNA to make mRNA, whereas translation is making mRNA into amino acids or a protein. And really, we're talking about a chain of amino acids. It's lots of amino acids connected together by those peptide bonds.
Apparently you don't need
Transcription happens inside the nucleus. Translation happens inside the ribosome. And so know the location. Remember that the reason why we need that mRNA is that transcription is happening inside the nucleus, the translation is happening inside the ribosome. And then lastly, translation uses that tRNA or that transfer RNA. All right, let's try a practice problem. It says, which statement best describes translation? So, keyword best. We're looking for the correct or right definition of translation. So, read over these statements, try to figure out what the answer is for these questions.
You know how ridiculously helpful that is?
All right. So which statement best describes translation? The process of making two DNA molecules from one DNA molecule. Wait, what? This is something completely different. This is actually a DNA replication. So this is not the correct answer. The process of making two new daughter cells. Does anyone remember what this process is, making one cell into two cells? Starts with an M. Mitosis, love it. So this is mitosis, not the right answer. The process of making mRNA from a DNA strand. What's this process? Is it translation? This is transcription, exactly. This is transcription. Key thing, we're talking about making mRNA, which is a clue that it's transcription. And then lastly, the process of making proteins. So translation is all about making proteins. We're decoding that mRNA strand. The best answer is D. All right, so D's the right answer there. Next one. Read over these statements. These are the events of translation. Put them in the correct order. So put these in the correct order. Looks like you have a couple different options. Figure out what thing happens first, what thing happens last. So when I read over these statements, I remember back to that video. The very first thing was that yellow mRNA, leaving the nucleus. So the very first thing is right here. So 2 is going to be number 1. The second thing was that yellow mRNA was matching up with the ribosome. Okay, so the mRNA matching up with the ribosome, okay, that's number 1. The mRNA is matched up with the ribosome. Now those like little butterfly tRNAs are coming in, they're bringing in the correct amino acids. So now it's number 3. As it brings in those correct amino acids, chains are being formed, bonds are being formed, those peptide bonds, number 4, and then number 5. So we're talking about 2, 1, 3, 5, 4. 2, 1, 3, 5, 4. The answer is C. All right. So we're on to the last objective, but just like the previous objectives, I want you to spend some time turning this objective into a question. So what happens? So use this into what happens. Turn this into a question. What happens? And the location of each step of translation. So answer that question in the objective. Use these objectives to quiz yourself. If you know the answer to the objective, there's a good chance that you really understand this information. So quiz yourself. Go back and check your notes if you need to. You just want to summarize what happens during translation. All right. So what's the location of translation? The location is at the ribosome. If you want to be a little more specific, it's the ribosome in the cytoplasm, because it's outside the nucleus. And what's happening during each step? Basically, we are making chains of amino acids. And in order to make those chains of amino acids, we have tRNA brings correct amino acid to mRNA in ribosome. Okay. This is really what's happening. We're making these big chains of amino acids. Those tRNAs are bringing in the correct mRNA the correct amino acid to the mRNA ribosome. We end up with these big long chains of amino acids. All right. Last objective. Read mRNA and use either the table or wheel to decode the amino acids. Okay. So now we're at the last part. We've already talked about how proteins are made of amino acids. Let's talk about let's practice reading that table and decoding it. Remember before we talked about codons? Codons are in groups of 3. So if this question asks how many amino acids are there, we want to split this up into groups of 3. We're talking about 6 different amino acids. And so we're going to separate them out, if that helps. And now we want to actually use the wheel, or use the table, to decipher what amino acids they are. All right, so use this table, and in your notes, figure out what amino acids are actually in there. So like, for example, the first one was AUG. We have A, we have U, AUG, methionine. You don't ever need to memorize this table. You need to memorize how to use this table. You will always get this table on questions, but you want to make sure you know how to use it. So practice using it right now. Looks like you guys are figuring it out. So we have AUG is methionine, GCU is alanine, UAC is tyrosine, CUG is leucine, UUG is also leucine, UGA is like the stop codon, or we stop making it. Technically, stop is not an amino acid, it's just to stop making them, but it's good to know what it does either way. All right, so they have another table that in my mind is actually a lot easier to use. It's the wheel. And the way the wheel works, let's say we have AUG. We start with the A, move to the U, move to the G, methionine. Much easier, right? We have UUU. We start with the U, move to the U, move to the U, phenylalanine. You guys like that one better? That one's on some of the problems. I personally believe that this one's better, but honestly, a lot of times in college they'll only give you the table, because that's what people are used to using. This one, in my mind, is a far superior design, because you just go with GGG, GGG. Glycine. So practice using that wheel now for this set of practice problems. So new set of problems, try using the wheel to see if you can figure out what those amino acids are. Okay, so use this wheel to figure out these ones. You start here. So you have like AUG, Easier? Yeah. I really do think this one's easier. So like if you have AUG, you start with the A, move to the U, move to the G, methionine. UGU, start with the U, go to the G, go to the U, cysteine. So you always start at the middle of the table, kind of move out to the outside edge of the table.
This is right. What time is it?
Probably switch now. Jordan, you can just go back and check in with your teacher to make sure they know you're here, and that should be fine. All right. Here's the answer key for those problems you just did. You should have methionine, cysteine, leucine, asparagine. This is my favorite amino acid. This is actually an amino acid that's in asparagus. Makes sense. It's asparagine, glycine, and stuff.
What?
Okay. So just to summarize some information before we do the last two practice problems. Proteins are chains of amino acids. So each codon or each three-letter sequence gives us an amino acid. We use those three-letter sequence codons to figure out what the big chain of amino acids is. 3 bases equals 1 codon equals 1 amino acid. Just have that ratio in your mind. 3 bases, 1 codon, 1 amino acid. And then just remember that we use the table or the wheel to kind of translate or decode the mRNA to figure out what information is in that mRNA. All right. So let's do some practice problems. Determine the amino acid sequence from the following DNA sequence. Really important vocabulary word here, guys. We're talking about DNA. This is the DNA. Before we can use this table, we have to turn this DNA into RNA. So make sure you turn this DNA into RNA first before you go to decode it. Okay. We have AUG, CUG. AUG is methionine. CUG is leucine. So C is going to be our best answer choice. All right. We are almost done. The very last thing we want to do is the last objective. I just want you to summarize how to use the table or wheel to decode the amino acids. So just describe, pretend like you're telling someone who's never done this before. How do you use that that table or that wheel to figure out the amino acids? Pretend like you're telling someone who's never done this before in their life. Give them some tips or some pointers on how to use these. What do you want to do? Yep. Yep. The third one. Yep. You start here, you down here, and then figure out find everybody. Did you miss these notes? Okay. I'll email the PowerPoint so you got access to it. That sound good? All right. Thank you all for coming. I will post this video as well as email you guys the PowerPoint.